Okay, class, so here we come to another important lecture, and this is the second step of the urine formation, and we refer to this as the selective reabsorption. And by the selective reabsorption, what we mean is that this is a process where the useful substances that we earlier produced here in the glomerular filtrate, so they would be reabsorbed back into the blood plasma. For those who have missed the previous lecture of the ultrafiltration, so you see that here we have the glomerular capillaries, and over there we had the blood plasma, and in the blood plasma, when we were trying to be removing the waste materials inclusive of the urea, the uric acid, the creatinine, so along with them, most of the water, all of the glucose and all of the amino acid and most of the vitamin minerals and the useful stuff, it also got filtered out from here from the glomerular capillaries, and here they have made a filtrate here, and that filtrate was the glomerular filtrate. Now, since the glucose and the amino acid and the vitamin and the minerals, they are the normal need of my cell, so what I would do is that I would cause this filtrate to flow in the proximal convoluted tubule into the loop of Henle and into the DCT and the collecting ducts, and at every part along the nephron, I would try to take the useful materials back into the blood plasma so that the composition of the blood plasma should not get disturbed and the normal activities of my cells they should continue to work or function properly. So, if I were to recall some of the parts of the nephron, so here this is the proximal convoluted tubule or the first coil tubule. Here we have this loop of Henle coming with us. Then here is the distal convoluted tubule, and then toward the end of the nephron, now here we have got these collecting duct, and we have to be understanding for today is that how come the selective reabsorption is being accomplished in these parts of the nephron. So if I were to be quickly telling you that the site of the selective reabsorption in the nephron, so this would be the first coil tubule and the PCT, this would be the loop of Henle, then there would be the second coil tubule or DCT or the distal convoluted tubule with me, and at the end we would have this collecting duct with us. And it is important to know that when we try to be selectively reabsorbing the materials out of the glomerular filtrate, so there would be the involvement of this diffusion process and the osmosis process and the active transport process, we say. So here, this is a quick summary that what is getting reabsorbed where. So here we have done the ultrafiltration. Here we have this proximal convoluted tubule where we would reabsorb most of the all of the glucose, all of the amino acid, most of the water and mineral and the vitamin, they would be reabsorbed here in the PCT. And the glucose and the amino acids specifically, they would be reabsorbed by the help of the active transport. When I come here into the loop of Henle, so here we would absorb the ions and the salts by diffusion, and then here the water would be reabsorbed by the osmosis process. In the distal convoluted tubule, we would also try to be removing some of the ions also back into the blood. And from the collecting duct, we would be also reabsorbing the water out from here, and then it would go into these blood capillaries, and that basically involves influence of the hormone that is antidiuretic hormone, we say. Now, here we would begin with that how come the mechanism of the reabsorption would take place here in the first part of the nephron, and this was a proximal convoluted tubule. So for the proximal convoluted tubule, you must know is that here all of the glucose, all of the amino acid, and most of the water, the ions and the vitamins, they would be reabsorbed back, and surprisingly, 50% of the urea would also be reabsorbed from here in the PCT. Now, if I were to be telling you the structure of the PCT, so here you see we have got the walls of the PCT, and here we have these cells of the PCT with us. Here is this empty space within the PCT where the glomerular filtrate is actually flowing, and this PCT is surrounded well by the blood capillaries, so that we should be taking the useful material from here and then again back into the blood capillaries that are surrounding this first coil tubule of the nephron, we say. So here, this is not the part of your syllabus, but we must be aware that how come the cells that we have here in the proximal convoluted tubules, say they are adapted for this reabsorption process. So I would take you now to the structure of this one PCT cell that I have got. So in this proximal convoluted tubule cell, we have these abundant mitochondria. We have these transport proteins for the movement of the glucose and amino acid, and these cells they also have the microvilli that would increase the surface area to the volume ratio. Now, if I were to be describing this, the structure of the proximal convoluted tubule, so we say that they have the specialized cells, they are the PCT cells, and they're adapted for the reabsorption process. And if we just look on toward the feature of these cells, so these microvilli, no, the word is not villi, but rather than these cells, they have the microvilli, and they would increase the surface area to the volume ratio for the reabsorption in the PCT. We have these abundant mitochondria that would release energy for the active transport required for moving the glucose and amino acids back. And then there would be the presence of these transport proteins as well, because they are specific to be taking one specific molecule out of the filtrate flowing in the lumen of the PCT, and it would be carried back here into the PCT cells. So now I am like linking this cell with the filtrate flowing out. So this yellowish is the filtrate. Here we have these PCT cells, and then surrounding them we have the blood capillaries. So if you were to see this in the filtrate, we have glucose, amino acid, the water and ions, and also we have got the urea. Here comes the PCT cells, and here comes the blood capillary. So I have to move them from here, then into these cells, and from here I have to take them back into the blood plasma so that the composition of the blood plasma does not get disturbed. So if you were to be understanding that how the selective reabsorption mechanism is taking place in the PCT, so you would say that these mitochondria that we have in the cells of the first coil tubule, so they would release energy in the form of ATP, and that would move all of the glucose and all of the amino acids back from the filtrate into the blood, we say, and some of the minerals and the ions they would also be reabsorbed from here. Now, the transport protein that we have got, so they would assist the movement of every specific useful thing that we have got in this filtrate. And at the same time, you see that when I have moved the glucose and the amino acid and the vitamin and the minerals back into the blood, so here the water potential would fall. So that would allow the water to move with the help of osmosis from the filtrate here, and then ultimately the water would be returned back into the blood plasma. Now, moving onward, we have got this loop of Henle with us, which was the third part of the nephron, and here the water would be reabsorbed by the osmosis, and the ions would be reabsorbed by the diffusion process. And if I were to tell you about the second coil tubule, so here some of the ions would be reabsorbed back. And now comes an important concept in case of the reabsorption of the water that we have got in the last part of the nephron, which was a collecting duct. So you see that the collecting duct has got again the urine flowing in it, and in the urine we have got most of the water. And that water, if I would take it back into the blood capillary, so this would not simply be carried out, but rather than there is involvement of the hormone that we have with us, and this hormone is entitled as the antidiuretic hormone, we say. So in terms of the reabsorption in the collecting duct, what we say is that here the water is reabsorbed, but under the influence of the hormone, and that hormone is antidiuretic hormone. And here is a note written that the selective reabsorption of the water in the collecting duct is an aspect of a homeostatic control, and we call this as osmoregulation. You see, by the homeostasis, what our body do is that it would try to be maintaining the internal environment constant of our body, like the blood plasma environment and the environment within the tissue fluid, in context of the temperature, in context of the level of the water. So this would be maintained by the homeostasis. So the water reabsorption here in the collecting duct is an aspect of homeostasis that we are specifically referring to this as osmoregulation. So here, if I just quickly take you to the diagram, so the scenario is that here we have got the filtrate flowing, or we have got the urine flowing in the collecting duct, which has the water in it. And the water has to be taken back into the blood capillaries, but it cannot come back into the blood capillaries simply, but rather than we want a hormone coming toward the collecting ducts that is antidiuretic hormone, and it would assist in the movement of water from here the space that we have in the collecting duct and back into the blood capillaries that we have got surrounding this part of the nephron. Now, we have to actually create a scenario, and the scenario would be that in some cases, the level of the water in your blood plasma, this would fall below the set value. And when this would fall below the set value, then here in the hypothalamus or in the brain, we have got this hypothalamus, and in the hypothalamus, we have these osmoreceptors. And these osmoreceptors, they basically detect that the level of the water they have fall in the blood plasma. Now, what this posterior pituitary gland is gonna do, that this would cause the release of the antidiuretic hormone. And that antidiuretic hormone, we would now carry it toward the collecting duct of the nephron. And when the ADH is carried to the collecting duct of the nephron, then this ADH would compel the water to move out of the urine back into the blood. And as a consequence of that, we will produce less volume and more concentrated form of the urine, we say. Now, if I were to just take you to this diagram, like after we got our blood plasma, water decreased to a very minimum value. So osmoreceptors, they compel the posterior pituitary gland to be making this antidiuretic hormone. This antidiuretic hormone would come and this would bind to the receptors that we have got in the collecting ducts. And when this would bind here, then ultimately the water would move out of the urine here back into the blood capillaries. And by that, the urine produced would be in less volume and it would be concentrated. So now comes the second situation. There was this random day and you had a lot of intake of the fluids. So that means that now I would like that the water should be lost in the urine and it should not be reabsorbed back into the blood plasma. So simple thing is that when the water level in the blood plasma it would rise above the set value. So again, in the hypothalamus of the brain, the osmoreceptors would detect it. And now they would cause this, you would say, posterior pituitary gland to not release antidiuretic hormone. So when the antidiuretic hormone is not released or less released, so it would not be taken to the collecting ducts of the nephron by the blood plasma. So simply, when no ADH is coming to the collecting duct of the nephron, so water would not be reabsorbed back into the blood, and as a consequence, the urine would now be produced in more volume and it would be dilute. In the previous situation, it was produced in less volume and it was concentrated because we compelled the water to be moving back into the blood plasma. So you see here in the last part of the nephron, we have collecting ducts, and in the collecting ducts, you see the urine is flowing and it has got the water in it, isn't it? Now, since already the level of the water in my blood plasma is high, so I would not cause this antidiuretic to come here and bind with the receptors of the collecting duct. And as a result, the water would not move out of the urine here in the collecting duct of the nephron, and the urine produced would be in more volume and it would be dilute. So I hope you would be clear with this important concept of the selective reabsorption that happens in the nephron.